probably usually get. Make sure that we're not muted. Oh, good.
Okay.
So maybe swap places. I'll introduce you real quick and then we'll get started.
Okay.
This is our great. Well, welcome to our CVRTI Seminar Series. First off, before I introduce our speaker, next week we have Jose Jalife will be here from Spain. Again, a pioneer in the field, someone that's very influential. We'll be excited to have him be our speaker next week, but encourage everyone to participate again next week. Today we have with us Ed Dibella, and I'm delighted to have him here with us. When I got to the U about 11 years ago or so, Ed was one of my first collaborators. We worked on projects right off the bat. Okay, may have some technical issues. Oh, now we're good? Okay, all right. So by way of introduction, Ed did his undergraduate degree, looks like at the University of Virginia, electrical engineering, master's at the University of Vermont in biomedical engineering, and then his PhD was actually electrical engineering at Georgia Tech, but with imaging technology stuff. And then he came to the U of U as a postdoc, and so he's been at the U for a few years now. Rose through the ranks, postdoc instructor, assistant professor, associate professor, full professor in radiology. He's the director at UCAIR, Utah Center for Advanced Imaging Research. As I mentioned, on a more personal note, he just is a terrific collaborator, somebody that I helped mentor me and help me out as I was getting started here at the U. So we're just happy to have him here. He's going to talk a little bit later. He's got a new grant that he's a multi-PI grant with Ravi Ranjan, the CVRTI investigators, is one of his main collaborators on that. And so he's worked with a number of investigators at CVRTI and all across campus. But with that introduction, I'm excited to hear Ed's talk today. Thanks, Ed.
Well, thank you, Derek, and thank you very much for the invitation to talk here. It's been a while since I've been in this room, but I've attended talks, and I think I've presented here a long time ago. I'm really hoping, I don't know who is in the audience, really. Maybe I can, okay, I can sort of tell. It doesn't show up there. All right, but please feel free to interrupt me, ask questions. I guess, is it easy for one of you to monitor if somebody raises their hand or something like that?
I've got the chat window open here on my computer.
Oh, okay, super. Actually, yeah.
I'll do a line as well,
Yeah.
Okay, great.
Yeah, because I have a whole ton of stuff I can talk about, but I'm really mostly interested in talking about stuff that may be somewhat relevant to some of your research. So I know CVRTI, there's a lot of electrical mapping, there's a lot of stuff at the cellular and molecular level. I'm usually imaging at a higher level, but maybe some of those tools, especially like perfusion, fibrosis, oxygenation, maybe some of those measurements can help or be interesting in some way. So that's what I was planning to talk about, but please, try to have me focus on things you're interested in. I'll start out with an introduction to UCAIR, Utah Center for Advanced Imaging Research, which used to be MERL, Medical Imaging Research Lab, a long time ago. Like Derek said, I've been here a while, came in 1995 as a postdoc when it was MERL. But we have approximately the first two rows, about 16 PhD faculty right now. And then there's about 16 MD or MD PhD faculty that also do research in the Radiology and Imaging Sciences department. We also just got a new, or our first ever vice chair of research. Tran Nguyen came from University of Washington. And so we're sort of in a growth phase. We plan to be adding faculty, making more investments in research. It's an exciting time. This is just a little graphic to show we have a lot of scanners. So 8 1.5T MRIs, 8 3T, 8 CTs, digital X-rays, SPECT CTs, PET CT scanners, over at Huntsman's, IR suites, ultrasound, mammography. It's a big operation. And all can be used for research, but mostly most of the research happens on dedicated MRI systems at the building my office is in, nearby in Research Park, Imaging and Neurosciences Center. UCAIR has a lot of research going on in diverse areas, from cancer to neuro to cardio, musculoskeletal, body, and AI. And there's actually relatively new AI initiative in which we've hired a data scientist, probably going to hire another faculty that more specializes in AI. I mean, different people in UCAIR do work in artificial intelligence. I mean, we have some papers in it, but others don't, and maybe can be boosted from people doing that kind of work, or maybe can continue very well without that. Funding goes up and down. This just gives you an idea of approximately how much annual grant funding we're getting, or foundation funding, on the order of 8 million in a good year, less in other years. Had quite the conversation yesterday, right, about Bremer ranking. And you see we hit 19th in 2019, but again, that's something that fluctuates, and there's certainly, an asterisk with Bremer and how that's calculated and what goes into it. But it's easy to report. And so I think my last UCAIR slide is just from our last in-person symposium, where we get together and invite people from outside the department, but it's mostly collaborators within radiology that have research interests and meet either on campus or somewhere else for a symposium. Okay, so I'm going to start off talking about myocardial perfusion. And why do we care about perfusion? Well, the biggest motivation is for coronary artery disease. Millions of people with chest pain go to the doctor and often get an imaging test, sort of a gatekeeper, before they would go to cath to try to figure out, is it really a heart problem? And often send people to SPECT, single photon emission computed tomography, and do an exercise test or stress test for vasodilator and see if you can detect ischemia. That's sort of a big gatekeeper. I've read there's something on the order of 5 million scans annually in the US for SPECT cardiac perfusion. Ultrasound is certainly a mainstay. Lots of people get ultrasound for a variety of things. MRI is a fabulous technique for doing it. So PET is quite good too. The PET has relatively limited availability. And so another reason I listed here is becoming better known, coronary microvascular dysfunction, where you don't have obstructive disease causing ischemia, but you have diffuse disease. So maybe this ANOCA, ischemia with non-obstructive coronary artery disease, can also be looked at with perfusion. And I guess I can just mention this graphic is, it looks like they're just trying to point out by visual assessment, the third case may have been called normal, but their quantitative assessment could pick up this diffuse microvascular disease and correctly call it abnormal. And then just trying to, mention something more related to CVRTI or more directly. I mean, there's an interesting paper. It talks about how the heterogeneity of myocardial perfusion is related to potassium channel expression and to action potential duration. And they actually were doing not MRI, they were doing microspheres to get their perfusion measurements. But I'll have a slide on microspheres. We do some microsphere work as a gold standard for perfusion to try to validate our MRI methods. So how do we do this MRI perfusion measurements? This will be really brief, but a really quick intro to MRI. So the signal that you get from the MRI basically depends on T1 and T2, rate constants that depend on properties in the tissue. And T1 is how fast your magnetization can regrow after you've excited it back to its normal equilibrium state. And that's usually around a second, 1,000 milliseconds in tissue and a little bit longer in blood, but does depend on your magnet's field strength. And this is a little graphic of it, taking a lot of liberties, but if you do an excitation, like right now, you've knocked the magnetization into this transverse plane where you can detect it. It's going around, you can pick up that signal with just a loop of copper wire, which you have placed around the person. And meanwhile, the magnetization, the green, is regrowing with rate constant T1 back to equilibrium. The red is losing magnetization, losing signal with rate constant T2. And we'll talk about T2 today also, but for doing perfusion with dynamic contrast MRI, you inject this contrast agent and it shortens T1, this gadolinium chelated to something so that it's not toxic, which it is in its native form. And it's a paramagnetic agent and will interact with nearby hydrogens to reduce the T1, to shorten it quite a bit. And it'll shorten T2 as well, but it's easier for the perfusion scans to look at T1 changes. And here it shows, depending on your concentration of gadolinium in the tissue or blood, you get very significant shortening below the 1 second. And so you'll get very different signals, and you can figure out what the T1 is from different areas and figure out the gadolinium concentration there and use tracer kinetic type techniques to quantify it if you want, or you can just look at it. So for example, here, we show a coronal slice there, and then we're going oblique or sort of perpendicular, so we get donuts, short axis slices in the heart, which is easier to figure out where you are. And so the gadolinium, if you inject it, will show up first in the right ventricle that's shown here, and then in the left ventricle blood pool, and then in the tissue. And you can see part of the tissue is not getting some of the gadolinium. It's not lighting up so much. Its T1 is staying longer. And then a little bit later, you can see that maybe even better. So here, there's some problem in the dark area, ischemia. And you do this both at rest and then again at stress being usually a vasodilator like adenosine. Some places do use a bicycle pedals, or there has been a treadmill, right? MR compatible treadmill right by the scanner, and then they do that to their maximum and then quickly get put on the scanner bed and put into the scanner. That hasn't really caught on, but it is it has been done a couple places, and you you usually try to get images each beat to see that gadolinium come in and wash out. And so you're triggered by the ECG, so you stay in the same heart cycle each time. You can cover 3 different slices, for example. And then you can just look at that and make diagnosis. That's what's typically done. But in our research, we'd like to quantify in milliliters per minute per gram perfusion. And so if you get an input function from the blood and you have tissue response curves, the signal intensity changing in your tissue, you can use a model and fit this as an input function convolved with, this is just a decaying exponential with the term that's related to perfusion, the term that's related to washout. And you can fit this data and get values for perfusion. So for example, here's a pixel-wise map to quantify flow in mils per minute per gram. So about 1 mil per minute per gram is sort of normal. And you can see there's this area in this case where it's 0. So it's actually scar or infarct. There's no perfusion going there. And we'll go into later, that's very fibrotic, dense scar. And you can do another kind of MRI image that makes it light up. And we'll talk about that. So I don't know, does anybody have questions or comments so far?
Don't see anything in the chat right now.
Okay. So anyways, like I said, clinically, you don't get that mils per minute per gram. And so you get hopefully pretty pictures that have decreased signal where it's ischemic. But that's really changing. And I mainly put this slide in just because I think it's a little different perspective on it's usually a lot of teams and different groups, I think, that change the field. And this person, Dr. Peter Kelman, a scientist at NIH, I think pretty much single-handedly has moved the needle on quantitative MRI perfusion, making it clinically viable and doing it in lots of studies. And he did it by, he has a wealth of experience in cardiac MR, but he wasn't really doing things in the perfusion realm. And I think he just, went to the problem, said what is needed, what is known. He contacted a bunch of leading people that publish a lot of papers in quantitative perfusion MR, like me, asked me some questions of like, why are there all these different methods and what's the right one and things like that. And they're questions that I don't think anybody could answer them. There are papers, with microsphere results, but it's just, there's so many pieces that go together, changing the acquisition so you can make it quantitative to what model you use, and there's an assortment. And so anyways, he came up with a model that he's made robust, even though it shouldn't work, it's not unique. And but they fixed that with some constraints. But then maybe a bigger thing is they added all of these QC steps that I would say most people just, especially like our group, we do 20 people, publish a paper, and then try to invent a better method, because that's our funding cycle, and that's often what is rewarded, when really we should probably do more like this and try to have a larger clinical impact. But so anyways, this is just a list of the QC steps, and the graphics don't really show up, but most people actually don't look at the triggering except when it's happening, and then it's usually just the tech there running it. And then the input function, I think they even throw out people that once you convert the gadolinium concentration, it should be a peak of 5 millimolar, very roughly, 2 to 10. And they probably throw it out if it's below 2. And they acquire a bunch of other information that's, just what studies are actually, they have confidence in. So that's a new development and is sure to spread. A little segue. So I'll talk more about atrial fibrillation because that's related to, both CVRTI and my interests. This was a paper with Derek and postdoc Nina a number of years ago where we were interested in quantifying perfusion in the left atrium. Can't do it with MRI because it's too thin. There's too much partial volume. You can get really high resolution sub-millimeter in MRI, but then there's so much noise. It just wouldn't. I mean, we can get numbers out, and I'll probably talk about that later, right? People have published not perfusion values, but for example, T1 values that I'll talk about later in the left atrium, and they got numbers, but I'm sure they're wrong. So anyways, this was just a review paper, and our thought for the left atrium was we really wanted to do it with microspheres because we're not sure that it's really that valuable clinically, but it would, in research questions, questions like, is atrial fibrillation partially caused because you're having bouts of ischemia that's stunning your myocardium, and eventually that becomes fibrotic, and then you have the conduction problems and AFib develops. So for example, and so we do have a microsphere set up, and this is a result from it. The microsphere setup is pretty neat device. Where the idea is you inject fluorescent microspheres 15 micron in diameter in the arterial side. And so they'll lodge in capillaries in proportion to blood flow. And so they're stuck there. You can do a few different colors because you're injecting millions of microspheres, but there's way more capillaries. So you're not affecting actual flow unless you were to use more or larger microspheres. And so you can use different fluorescent wavelengths, inject them at rest, at stress, at different states. And then after the experiment, so this is, highly invasive, these were mini pig studies. You can take the heart out and mount it in this apparatus here. And it basically has a blade that'll slice off. This is a cryomicrotome set up here. The blade is set up to automatically slice off whatever you dial in. I think 20 micron, 50 micron slices, so very thin, and the slices are just destroyed in our setup. But then it takes pictures with a camera and a filter wheel for the fluorescent, excites the fluorescent wavelength you're interested in, takes the photo, trying to actually see every microsphere. Now, most microsphere methods, you just take a chunk of tissue and you digest it in acid, and then you figure out how much fluorescent is there, putting it into some counter. If you could actually find every microsphere, you can do higher spatial resolution, you can get more information out. It's really valuable. It makes huge data sets and, runs overnight doing this slice and taking pictures. But that's what this is here from 3 different earlier states that just have subtle differences because they were all at rest, but with different microspheres. And then under adenosine vasodilation, where I guess I lost the color bar, but it's about 3 times higher, these flows. But you can see it's pretty heterogeneous, at least in this case, which is interesting. We've had a lot of struggle with getting good global scaling because you do need some reference number to scale absolutely your microspheres. What does 50 microspheres mean in terms of flow? You need to have a pump that pulls, and it pulls at 10 mils per second, it gets 500 microspheres. Well, then 50 is going to correspond to 1/10 of that. So you need something to calibrate. Okay, so I was just going to mention one other piece of perfusion, and then we'll move on to fibrosis. And this other piece is again sort of motivated by atrial fibrillation. Usually you gate the study, the perfusion study, and get 3 slices per R to R interval per beat. And what if your R to R changes? Well, that third slice, the way the scanner's set up, it's going to still acquire it, but it'll miss that trigger. And then it'll just wait for the next trigger. And so 1, you've got it like at a different cardiac phase, it'll look a little different. And 2, you've got this dead time. So both are bad. What if you just can ignore the gating, just acquire 3 images as fast as you want? And that's what's done here. We'll regular gated, and you can see the gadolinium injected light up and then lights up the tissue. And an ungated scan, where you can see it also beating systole and diastole as it goes. The images are acquired, just many more images. And then you can come up with schemes, for example, to try to self-gate it and group the systolic or diastolic together, or the same slice together, and deform them to do that. And you can come out with something where you have an ungated system versus a gated, and you've gated it to systole or diastole. And so this was really interesting, helpful work, especially when we went and tried it in people with AFib. Eric Beijing published this not that many years ago, and had good results. I think like I said, we usually do 26 AF patients, and had good sensitivity, specificity for detection of coronary artery disease, where the truth is going to be what you find at cath. And so that's really your goal of how well you can do that.
After that first pass, where does the gadolinium end up? You still see, you take images on a longer term. Do you see it recycle back?
Yeah, so that's a great question. I mean, basically those curves die out and things come to a steady state between the blood and the tissue, and then it's cleared by the kidneys, which is, I don't know the half-life, hours. So the perfusion, you really want that first part. That's what's driven by perfusion. The equilibrium part we'll talk about in fibrosis, because that's going to give you fibrosis information, and extracellular volume information. So it's a great question to segue to the next part. Yeah, but any other questions or thoughts or comments?
Yeah, I had one question on that. Could you do a blood draw, and then if you infuse a known amount of gadolinium and then do a blood draw after, can you get more information about total blood volume, and then also you'd have how much gadolinium per mil, so you'd really know flow per gram, right?
So good thinking. So the perfusion studies, you really want to convert to gadolinium concentration for your arterial input function and for your tissue curves, and that's pretty well established. We can check that in Phantoms if you set it up correctly, use the right sequences, stuff like that. If you added a blood draw, you could confirm that at some certain time point. Your gadolinium concentrations are going to be changing. If you wait till a late point, it'll be relatively stable, but even then it's changing, for a while, as it's just changing more slowly. So it could help you validate. It's not like in the microspheres where it's going to help you, where it's going to define your calibration. It'll just give you that one point that, yeah, you're converting to GAD concentration correctly, and that's useful. So most people, that might be a good idea to get validated initially, But then in routine use, I think you'd rely on your methods being accurate to convert to gadolinium concentration. So,
Yeah, I was just kind of thinking with heart failure, you have lots of different blood volumes, but depending on, their current state.
yeah.
That could be
So-
a challenge going different time points in their care.
So that's a great point, especially like, nobody ever talks about, can you do accurate perfusion in heart failure? I mean, say your ejection fractions reduce quite a bit and you inject this bolus and your perfusion relies on a pretty tight bolus, to figure out this kernel, you're fitting these parameters to get something related to perfusion. But if you're just injecting really slowly, which is essentially the same thing as having a low ejection fraction, you may not be able to define those parameters uniquely, and you may have inaccuracies. And while this has been mentioned in the literature, no one's ever, that I've seen, has ever studied and quantified, look, you really did get the wrong answer in this case. I mean, I suppose when you have a reduced enough ejection fraction, you're going to assume your perfusion is poor, and maybe you're not adding a lot to your study, and maybe you just, if you can see differences between different regions, but not quantify. So really good.
I had one follow-up question since you were looking at gating. There's been more attention with the microvascular perfusion, and pressures are really sensitive. So to getting the perfusion during diastole, because that's when the heart's relaxing and then there's less tissue compression. You any thoughts on trying to get that last phase of diastole before you even have stretch of the heart, to see what max perfusion might be while the heart is relaxed?
Yeah, so that's another really good point. There's been some studies to try to look just in normals, difference of perfusion at systole versus diastole across the cycle. And they basically find they can't tell a difference at rest. At vasodilation or stress, they can tell some difference. And something that they didn't probe, but seems like would be interesting in perfusion and things like oxygenation is how much is subendocardial versus subepicardial. Since, they squeeze different, you get more squeezing on that subendocardial side at systole. So your blood pool's a bit different. I think they're really interesting questions that are beyond the level of detail you need for coronary artery disease, but are of interest for a lot of other things. So yeah, thanks for the questions. Now we'll switch to fibrosis. And so SCAR is extreme fibrosis, and MRI is just magnificent at delineating SCAR. And because you can make normal tissue be black. And so abnormal tissue, SCAR in this case, really lights up. And I'll try to explain that. So you inject gadolinium. This slide is trying to show little gadolinium molecules as yellow dots. And, on a very high scale. So this is some sort of, capillary with red blood cells, and then the tissue. So gadolinium is extracellular. It doesn't go into any cells, but it'll go in the bloodstream and outside the bloodstream into that extravascular space. And, if you give it a little time, it'll be in proportion to how much extravascular space you have, which is related to fibrosis, having more space to distribute in, less cells that keep out the gadolinium. So you get more if you wait a little bit, and a little bit, I mean 10 minutes is pretty typical. And so after that 10 minutes, you're going to have some normal tissue that has gadolinium in it and has some T1 recovery here in in green. And then you're going to have some scar area where there's more gadolinium has distributed. So higher concentration, shorter T1, this black curve, it recovers faster. And so that's what's going on the recovery, but you can set up your MRI scanner to image at whatever time snapshot you want. What you do is you do this 180 pulse that I won't go into, makes your magnetization negative and then it regrows. But you know the time, for example, for normal to go through 0. So you just image at that time TI. You wait this TI time, and it's usually like 200 milliseconds, 300 milliseconds, depending on how much gadolinium, how fast, how it's distributed. And so you can zero that out, and now you get black for normal, and you can see you're going to get higher signal for something with more gadolinium in it. So that's how you image the scar. And this is called LGE, late gadolinium enhancement. It really is like a huge feature of MRI because, you know, you can see differences like this on CT and other modalities, but you can't crank the contrast like that so easily. And so it gets heavy use here at Utah and other places, for example, for atrial fibrillation management. So here's a transaxial slice showing the left atrium, and here it's labeled left atrium, left ventricle, aorta. And we can ignore this image, which is just showing where in case space for MRI you're sampling. And that's related to some of our new work. And then if you give, if you look after ablation, you know, you have these scar areas that are lit up here in white, bright, because you're using that kind of LGE sequence that tries to make normal tissue black or something like that. And so you can see quite nicely, you know, where exactly did you ablate, where did you miss, something like that. And so we'll go back to this, but first we'll go forward and say that LGE, while it gives you that great contrast, it's not quantifying anything. It's just trying to identify. And so if you could actually know the T1 values of every voxel or every region you're in, you know, you could recreate that kind of contrast. And so now that's much more the effort. And so people do T1 mapping. So you can, again, you can change how the MRI is acquiring the data. You can acquire it different time points along these curves, and everything's relaxing at some rate. Each voxel is, it's characterized by its T1. So if you have a couple points on it, you can fit every voxel to give you a T1. And so you can do that before you give gadolinium, after you give gadolinium, and that can give you the concentration in the tissue. Because if you know before and after GAD, you can figure out how much GAD there is, convert to concentration in the tissue and the blood, gives you this partition coefficient. And most people use a scalar multiple of that, the extracellular volume, that, you know, sort of tells you how much fibrosis, how much gadolinium is distributing. There's also been a lot of work nowadays in speeding up the T1 mapping method. And I just cite one of our recent works that is submitted down here that uses artificial intelligence techniques to try to go faster. Usually you can do one slice in a long breath hold. You can make that quite a bit shorter. And then the reason people haven't really done T1 mapping in the left atrium yet is the walls are so thin that to get a reasonable resolution makes everything so noisy that it's not very meaningful. But that hasn't prevented from actually, I know at least 2 papers that have published that, you know, you do get numbers. The 2 papers, the numbers are like at least factor of 2 different from each other. And they're both just trying to do a 2D slice, and they don't bother to even try to get really high resolution. They draw on the left atrium and report the signals. But it is a wave of the future. And so the last slide for this part is from a recent review paper on T1 mapping, which, this graphic came from, and this is a T1 map, but then it shows you up here all these different images from different people, what their T1 maps look like, including normal in the middle. But maybe of even more interest is once you compute the ECV from the pre and post T1 map, as you can see, for example, in athlete's heart, cells hypertrophy, and there's less space for the gadolinium. And so there's a lower extracellular volume than normal. And then all of these disease states have larger extracellular volumes than normal. And so it becomes a very powerful tool that is really starting to be used clinically. So actually, they do T1 mapping pretty routinely in a lot of their cardiac MRI, clinical cardiac MRIs here at Utah. But one of the problems, right, is there's just a lot of different things that influence your image. And it turns out T1 varies. Like even if you use the same sequence, you go to a different scanner and maybe even a different software upgrade or different day, and you get a little different T1. And so right now, people haven't really figured out to make it truly quantitative. So they say, oh, you should get, do a bunch of normals at your site so you know what normal is. And then you can much more confidently diagnose these diseases. So anyways, I think we're about at the stage of going to bring in 50 normals just to get their T1 values on our scanners and have more confidence versus using literature T1 values. Okay, so I think I have 4 or 5 slides now that are really related to this fibrosis and LGE applied to atrial fibrillation. And like Derek mentioned, we were fortunate to get this R01 funded, just started March 1st. So with PIs, myself, Ravi Ranjan and Eugene Komolsky, and a bunch of other, very talented people on the grant. And just, I'll run through quickly the motivations and the specific aims of the grant. And, maybe there'll be some synergies of work that others are doing that we could work into some of this funded project. And so anyways, part of the motivation is the left atrium is really thin, and there's these motions, especially respiration, really can reduce quality and add to scan time because you set up this navigator gated. So only when the diaphragm's at a certain spot in a certain window do you acquire your data. And so that can go on forever. And there's low contrast sometimes between fibrotic tissue and normal myocardium and blood. And the standard that pretty much everybody uses in all the big trials is not an isotropic spatial resolution. The slices are thicker than in plane, which causes some artifacts. And then it's very challenging to segment the LA walls, in part because the image quality is not always very good because of these motions and other issues. It is motivated to do these kind of MRI studies because they can be predictive of whether it's worth doing ablation in people, can be predictive of how well you'll stay in sinus rhythm. And some fraction, this is in really good sites that do a lot of them. You often have to drop 15% of quality that just aren't worth analyzing, not useful. And there's been in not very many studies, showing how repeatable this is. I will show one we've done in post-ablation. Post-ablation is a much easier problem, but pre-ablation is challenging. And so the first aim is to develop efficient and repeatable MRI methods. And so we've switched to isotropic scan, get rid of the navigator window. We'll navigate afterwards based on the MRI information. We'll make sort of sub images and figure out where we are. This odd graphic here is again this case-based. It's just MRI acquires, really Fourier transform samples, and which ones are you going to sample? You actually can throw a lot away and then use fancy reconstruction methods that use regularization, that use compressed sensing, that use deep learning, and do a better job from less data. And so that's this first aim. And really, Ganesh Adleru and Jason Mendez are the experts driving that new sequence acquisition and reconstruction. And then the second aim is to try to automate the segmentation part of the left atrium and the identifying what part of it is fibrotic. And this is really challenging because even experts right now, you have a big variability when you give them one of these images, especially if the quality is not in the excellent side, if it's just in the middle. And so there has been a lot of challenges, including the Utah database that Rob McLeod put together and makes available. There's been papers, people have applied deep learning methods to it and reported how successful they are. But I think we're ready for the next generation of better quality data and better knowing the truth. And then Shireen Alhabian at Ski and Alan Morris have a lot of expertise both in shape analysis and image analysis. And so we're going to have a lot of techniques that they'll apply to these kind of data sets. Then the third aim is to try to have truth. So to rigorously test and refine these methods in dogs that are AFib models. And the last aim to try to figure out the reproducibility in people and to compare it to the standard methods, so to add on both our method to what's being done clinically. This is just an example graphic that Robbie's group published showing that the repeatability between scan 1 and scan 2 is very good. These are all post-ablation. Pre-ablation, we expect much less scar, much more subtle fibrosis, and not as good repeatability. Okay, and last,
Can I
yeah.
ask a question? So on, if you were to do something, the atrial wall is so thin, it's a difficult target anyways, and fibrosis imaging is tricky. If you apply that to like left ventricular wall and you're looking at heart failure or other conditions where you get fibrotic development, is there ways to be quantitative about the amount of fibrosis, or is it still difficult to come up with ground truth, what's zero, and what's, can you be quantitative with fibrosis quantification, not scar, but fibrosis quantification in an easier target like left ventricle? How do you do that?
Yeah, so I think that's a great question. I think that slide that showed all those ECV cases, I don't think I really talked about, T1 can vary, you want reference, but when you're doing pre and post and you get concentration of GAD and then you compute ECV, those people pretty much agree on, and those are valuable. And so I think with enough pixels, with enough resolution, with the person holding their breath pretty well, those are really useful tools.
Pretty quantitative
right now.
at that point.
Yeah.
Okay. And solid, good MRI.
Yeah, they have pretty small standard deviations when those things are met. Some of those patients that it's impossible to hold their breath, or
Sure.
Twitch
when you're imaging or whatever.
Yeah.
Did you say for AFib you use dog models, right? So if used dogs on the model. I was wondering, has anyone tried using like cardiac perfusion to stop the heart and then using a stopped heart to make measurements? Like you said, the beating heart and moving.
I like your thought pattern. I'd much rather image, stopped hearts. Stopped hearts. I actually tried to switch to brain imaging because I'm like, oh, the heart's too hard, but the brain's too hard for me to get funded. I've been trying a bunch of things in diffusion. But no, as part of this study, we actually do studies after sacrifice. We leave them on the scanner, do sort of the best imaging you can, and then some of the hearts actually get imaged again. They get taken out ex vivo imaging to try to do diffusion of the walls. That's Eugene's nodding his head because Robbie's group does that. So it's a great idea. I guess you could start it up again too if you made the model that way. And the gadolinium circulation is gonna be a function of that. And so yeah, sounds good. So the last thing I'm going to talk about is oxygenation, which is a new area for me, but I think is really interesting. It's not a new area for MRI whatsoever. fMRI in the brain, functional MRI, I think is probably does more studies, at least research studies, than any other type of MRI. I mean, psychiatry, psychology, many questions that they're seeing what area of the brain lights up when you're thinking, because fMRI, what happens is the signal is proportional to how much deoxyhemoglobin is there, because the deoxyhemoglobin will make the T2 or T2 star be shorter, so you can pick that up on MRI. So if you're changing someplace in the brain, how much deoxyhemoglobin is, how much there is, which if you're using more oxygen in some area, that you'll get more deoxyhemoglobin, you get that signal. And the signal's not a big change. So the fMRI studies usually have a pattern. They'll show you something, stop it, show you, stop it, and then they'll fit some response to that and see where in the brain. Yeah.
Ade has a question. Ade Aromalara. Ade, do you want to ask?
Yeah, thank you, Derek. Yeah, so I think Derek essentially touched upon this this issue here. So I'm thinking in the context of the fibrosis. I'm assuming this is going to be a model of permanent AF. So I'm just trying to think. I'm thinking about the distribution of fibrosis. Is this going to be something like a patchy localization, or more of a global fibrotic distribution and stuff? And is this going to be a progressive development of the fibrosis? Or I don't know how this is going to factor into your model, essentially.
Yeah, so that's a great question. Actually, Derek can probably answer it better because he's been involved in a lot of models, goats and sheep, and very rapidly paced for a long time, and we still don't get very much fibrosis.
Right.
And it's a big model compared to humans, but it is something. You can develop some patchy fibrosis and quantify it with histology and get some sort of gold standard.
Okay, so then it would be fair to assume that it's not going to be like a permanent thing. It's just a persistent thing that basically transitions to permanent AF, or.
Yeah, I mean, in fact, for the imaging, we'll cardiovert, or I won't. Eugene will, and for the imaging sessions, take them out of AFib.
Sure. Right. Thank you. Yeah.
It does require chronic persistent AFib. I mean, long-term AFib. You can't just put them in AFib and then do imaging for a week. It's months or longer to develop any kind of fibrosis to be picked up.
Yeah. Okay, so back to the fMRI, and I'm gonna go sort of fast because we don't have much time, but this is just showing changes T2 and T2 star. And then here's an example fMRI study, which I just saw, which was just, too funny not to include. They give people a stimuli showing like this slide that says sandwich. The next slide either shows a sandwich or it shows, this is hard to see, but it's a couple bears that see people in sleeping bags. So you go, ha ha. And they're saying, okay, which part of your brain lights up when you see this one versus this one? What is humor? So again, they're just looking at two star changes with MRI to do this. And then I had to quote them just because I got a kick out of their first line of the conclusion. To conclude, no consistent conclusions about this have been drawn. So not the model of how to write your paper, but maybe it was in line with their, joking attitude. And so can we do cardiac fMRI? This is a much newer question, but this paper is still reasonably old, 2012. And so you can do T2 weighted in the heart. And now your stimuli is going to be do it at rest and then do it with a vasodilator like adenosine. And so you have rest and then stress. Doesn't really look like a difference, but they can compute signal differences in the tissue. And here's their values of how much it changed in percent. And you can compare it to stress perfusion where you see some ischemia in the subendocardial part of this wall. They can only image one slice because this isn't a real quick technique. This is, I think, 3 different people, but shows you get some correspondence. So it has promise to, what's the benefit? Here, you don't have to give any gadolinium. So that's nice, especially people with kidney problems. It's an issue to give gadolinium. Maybe for everyone, because now they've found it sort of sticks in part of your brain forever. They haven't associated that with any problems, but may not be the best thing. So this T2, what's going on? Why are we getting better signal? And that's a current question. So a nice study was done that relates it to perfusion and extracellular volume. When perfusion goes up, right, there's more oxygenated blood. Probably deoxygenated is less as a percentage. So you're going to get longer T2s. And then if you have a larger blood volume, like with this vasodilation, you have more blood there, but you're using, if you have the same cardiac work, you're using the same amount of oxygen. So again, the deoxygenated hemoglobin is less. And so those are some of the ideas of why it's changing. And then there's a neat idea to go to needle-free. So don't have to give gadolinium, don't have to give adenosine, which actually is a pretty huge effort and a risk right now, because like we have to do 2 separate IVs. We can't, the adenosine has to be an infusion. Your GAD wants to be a bolus, so they don't work well together. And it's a pain, needs cardiologist coverage to give adenosine. So here's an idea, you just do hyperventilation for a minute. That leads to vasoconstriction with the blowing off carbon dioxide. And then you breath hold and image, and you start vasodilating during that. And so you can see a sequence of changes like this in time. Here's a normal, and their signal is increasing because of the T2 is getting longer because there's less deoxy hemoglobin. Essentially, when you're vasodilating, this is during the breath hold, you've hyperventilated up to time 0, and then you're doing the breath hold and it's increasing. But in ischemic areas, you see a different trend. And so this could be really valuable. And then I'll skip, there's fancy ways of trying to do this. And so this will be the last thing. You could also add in strain. So strain comes from, actually these oxygen sensitive scans that we've been doing are while it's beating. So you're getting systole, diastole, and in between phases. So from those slices, you can also calculate strain. Strain's just deformation of the slice. And so this is color coded the strain in a normal up here. How the strain changes and how the oxygen sensitive changes. And then in an abnormal, you see different patterns of change in the strain and the oxygenation. And this is a very recent paper, but they actually found, we'll just look at the oxygen sensitive, gave a large area under the curve. This is for sensitivity, specificity of predicting coronary artery disease, actually matching with the Israel region. They had post-stenotic regions and remote regions. But the peak strain didn't do as well, but put them together, and that does a little bit better than one by itself. So last slide, cardiac MR tools are improving, especially if you have applications that could benefit from perfusion, quantifying fibrosis or quantifying oxygenation. There's a lot of interesting questions with interplay with arrhythmias, NEP, and with thin walls and high spatial resolution. And I hope you can take something away from this talk, something valuable for your own research, or that might lead to new ideas in the future. So thank you.
Thank you, Ed. We're at about the time now, so if people need to go, other meetings or whatever, start logging off.